The aim of the following experiment is to study the dynamic processes of materials in liquids in real time with high spatial resolution using transmission electron microscopy. This is achieved by first microfabricating a liquid cell from ultra-thin silicon wafers. As a second step, inject 100 nanoliters of reaction solution into the reservoir of the cell using a syringe and Teflon nanotubes. Then, seal the liquid cell with a cover. Next, load the liquid cell into the TEM sample holder as a standard TEM sample, and insert the sample holder into the microscope for in situ TEM experiments. This process shows the dynamic growth of nanoparticles in liquids, such as the formation of platinum-3 ion compound nanowires using real-time imaging with sub-nanometer resolution.
The main advantage of this self-contained liquid cell technique is that a thin liquid layer can be maintained in the viewing window long enough to allow an extended chemical reaction to occur.
This method can illuminate key aspects of material science and physical chemistry, such as growth and transformation dynamics of materials in liquids.
This method can provide insights into the nanocrystal growth and assembling mechanisms. It can also be used to image biological materials in their native environments.
Generally, individuals new to this method will struggle because it requires a series of fabrication processes to make the liquid cells, and it's a challenge to handle the tiny liquid cells for the in situ TEM experiments.
I have always been fascinated with how crystals grow.